Good morning or good afternoon, depending on your time zone. Welcome to the Harmful Algal Bloom webinar series. My name is Amy Weckel, and I'm from the Illinois Water Resources Center at the University of Illinois at Urbana-Champaign, and I'll be your moderator today. This webinar, which I'm trying to advance the slide here, 1 second. There we go. This webinar is hosted by the Algal Bloom Action Team, of which Illinois Water Resources Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resource Research Institutes, the North Central Region of Water Network, and university extensions within each state in the North Central region. In addition to hosting a webinar series, our team has developed a website of resources, including fact sheets and frequently asked questions about HABS topics. I encourage each of you to visit our website, which I'll put in the chat. And there you can explore some of our resources we have to date. 1 second, I'll put that in. Go. Including what you should know, you should know fact sheet, a HABS frequently asked questions database, and recordings from previous webinars and our virtual HABS research symposium, which has been held each January for the last 5 years. Before we get started, just a few housekeeping items. We will have a Q&A session after our speakers present today. So please list your questions for our speakers in the Q&A panel, and we'll do our best to address all of them following the presentations. If there's a question already posted to the Q&A panel and you have the same question, upvote the question and we'll be sure to get the answers to the most popular questions during the session. Today's presentations are being recorded and will be posted on the Algal Bloom Action Team website following today's session. If you're having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat and we'll be happy to assist you as best we can. Today's presentations focus on cyanobacteria in the Great Lakes and other large lakes, and we have 2 great speakers lined up for you. Our first speaker is Richard Stumpf, who leads NOAA's efforts to translate forecasts of harmful algal blooms from research to operations. He also tries to make satellites useful for coastal environmental problems, such as figuring out how deep the water is and finding pond scum. Stumpf has 40 years of experience in coastal oceanography, with particular interest in harmful algal blooms. He has developed methods to use satellite data and modeling to understand coastal eutrophication, habitat and wetlands change, shallow bathymetry, and algal bloom monitoring and forecasting. He received a BA degree in the environmental sciences from the University of Virginia and MS and PhD degrees in marine studies from the University of Delaware. He has worked on all the U.S. coastal and Great Lakes states except Connecticut, Georgia, Oregon, and Minnesota. Very impressive. I'll stop sharing now, and Richard, you can take it away. You are muted.
Okay, thank you so much. And Alad, I'm working on getting to those other states. The 4 I still haven't worked on. So I'm going to talk about some work we've been doing to develop monitoring time series for the Great Lakes, but also applicable to other lakes in the country. And example here is this is Western Lake Erie you see here from satellite. I have a number of colleagues who support this effort, but one who's done a bit more work in this is Sachi Mishra, who I mentioned here. And we have a lot of support from the Great Lakes Restoration Initiative for the work we're doing here. Oops. Are you still sharing the screen? No.
No.
Okay, hit the wrong button. All right.
There we go.
This should work now. Okay. So our work started in Lake Erie, and 2003 had probably what was described as the most severe bloom in Lake Erie's recent history. That's not an image we generated, but it was a common one at the time. We started in 2008 to try to see what we could do with some of the new satellites, and happened to hit what is probably the worst bloom Lake Erie has seen, which was in 2011. That's distance, it covered well over 150 miles of shoreline at its peak at this time. So our goal was we developed a method to detect the blooms. I'm not going to go into details on the method here. Um, and um, that work we've done in Lake Erie has in identifying cyanobacteria blooms specifically from satellite, we had a method that was broadly applicable to many lakes, and that led to the interagency Cyan project, the Cyanobacteria Assessment Network, and developed with EPA, NASA, USGS, and ourselves. And there's many pubs on that. You can get more information about it at the site. And that supports detection of about 1,800 to 2,000 large lakes, depending on how you do the math with these. Example result that I show here is changes from what was the Maris time series from 2000 to 2016. I'm sorry, 2002 to 2012 was when Maris was up, and comparing to Olchi, which started in 2016, and what were the trends between those? And the red points are lakes that showed a decline between those two time series. Blue show an increase. Now, this was done with 4 or 5 years of Olchi data. And keep in mind, when you see this, it looks like things are getting better, but there are cyclicity in these. And some of the other pubs have shown that Olchi has been showing a trend towards potential decrease over time. So there are always fluctuations. Nothing is unidirectional, going in any one direction or the other. But it does give a sense of this, at least on a broad scale, from one decade to another decade. Some issues here, if you try to look at in more detail, is the Maris. Unfortunately, the satellite failed catastrophically in April 2012, and so there's no data set from these two instruments for the summer 2012 through 2016. And the Maris 300-meter data is also gappy. The 300-meter was collected by request for an onboard tape recorder, and there's a limited amount that could be collected on any overpass. And so it was not collected as frequently as it could. The lower resolution it's about 1.2 kilometer Maris, was collected routinely around the country. So that provides a more complete data set, but a lower resolution. For Lake Erie, we did develop a time series with the 3 satellites. So we used the Maris 1.2 kilometer. We did a gap fill with MODIS. I'll talk a little bit more. The MODIS sensor that's a NASA Terra satellite. It has some bands that allow it to match pretty well to Maris and to Olchi, but it has some issues. And then when the Olchi sensor came on in the Copernicus program on Sentinel-3, we switched to that, and it's the equivalent of Maris, and it's well designed for the satellites. Now, so this time series, you can see some of the severe blooms, 2011, 2015, and they vary quite a bit in Western Lake Erie. Of course, the satellites aren't the same, and just give you a perspective on what the sort of difference makes between 1 kilometer and 300 meter. So at least face level, these are the same date. Within these 2 passes are in less than an hour from each other in time, and the basic true color MODIS is capturing the bloom perfectly fine for Lake Erie. But at 1 kilometer, you can see how blocky it is, and you really start losing detail close to shore. And then certainly in smaller lakes, you would lose ground. But at least in a larger lake, or like Sandusky Bay here, you could pick up data pretty well at 1 kilometer. But just a short detour to get an idea of how the resolution matters. You really need at least 3 pixels as a minimum width for any water body in order to work with. And the reason is you need the pixel to only include water. As soon as you have a mixed pixel, like this one here, I assume my cursor can be seen okay, that the lakeshore goes through, you have vegetation in here. And as soon as you put vegetation in there, which is much brighter than water, you end out with a real problem trying to extract meaningful spectra. So you need enough pixels to allow for mixed pixels on either side. So that gets to that. So what it means is If you have a 300-meter pixel, you could potentially go to a lake as small as 1 kilometer wide. 1 kilometer pixel means you have to be greater than 3 kilometers across. That also doesn't account for any noise or residual response of the sensor, but that's the absolute minimum you can work with. To get another sense on how these work, there's a color coding here, and if it's green, that would be best quality. The pale yellow is good. Orange is adequate. And Algae is certainly for large lakes. It's really the best sensor. Spatial resolution would be great to get a little higher resolution so we could push down into smaller lakes. There's tens of thousands of lakes in this country, and that Algae cannot pick up, which of course there's a great interest in. But at least for those large lakes, it's okay. Key thing is on the temporal resolution, how often you get a pass during a week really matters. And the 3A sensor went up in 2016 and 3B in 2018. And if you have 3 a week, that's okay. But we're up to 6 a week with 2 satellites, that really makes a difference in the data quality. Our ability to clear clouds, we can't see through clouds. There's also the potential with cyanobacteria about wind mixing of the blooms. If we have microcystis or dolichospermum, the ones that are buoyant and form scums, we see them very well on a calm day, and the satellite measures surface concentration, not the total through the water column. And so if you have wind mixing, that's diluted the concentration of the bloom. The biomass is mixed through the water column, so you don't necessarily see it. So you really want more images to get clear and low wind during the time. It has the right combination of spectral bands. I could give a whole seminar on how that works, but in order to pick up cyanobacteria specifically, Maris had the same bands. A definite trade space is on the 300 meter is the frequency, as low as only 1 per week for some of this time period. Once we're at the low resolution, it's up to 2 or 3 a week, which is okay. Be good if it's better, the right bands. MODIS is 3 to 4 usable images a week. It actually collects more data. As far as how far in, rule of thumb is about a Secchi depth. If there's scum, a Secchi depth might be 1 centimeter or 2 centimeters, but that's kind of the rule of thumb for how deep into the water. MODIS, it actually has more passes, but it points way off nadir. And so your pixels start becoming 2-3 kilometers across, and you're looking through a lot of atmosphere as well. And so they become unusable. So really you get about 3 to 4 a week with the Terra sensor. And the bands, some of the bands saturate as well in bright targets like in blooms. And so that requires some additional special processing to try to get the right answer. So it's a good instrument, but it's just a little more awkward to deal with as well. And again, clouds can take out a half to 2/3 of imagery. Terra tends to get sun glint. We have less of a problem with Algae on that regard because of the way it lays out. And our algorithms do well with that. So there's kind of a crash summary on how these sort out. Now we developed this as a running time series. So we were doing live monitoring for Lake Erie, and each year we would add on to our time series. Well, so our last reprocess was 2012. Maris got recalibrated a few times. MODIS, with the saturation, we ended up changing our approach to how we deal with the saturated bands, using other bands as a compromise. And so we did it one way in 2012, a little different in 13, a little different in 14. There's also, these are old satellites, 25 years old MODIS is old at 25 years, and it's showing its age. There's decline in a bit noisier and a bit of decline in quality. Algae, as we were doing this, we started off with the calibration that was done in the laboratory before the sensor was launched. And then there's been post-launch calibrations and also a change in atmospheric correction recently, which affected a key band we use for cyanobacteria. And so we do not have a consistent data set of what we've developed as a time series. And if you're going to do analyses over time, you really need that. So we developed a method and strategy to update the time series. And with Maris, took in all the calibrations. We've also picked up all the data, which we didn't actually have at the time. Some of it took a while to collect, and we picked that up. And we also intercalibrated Maris Dolce. Keep in mind, they didn't overlap each other. So we actually used the MODIS where the bands were comparable, took advantage of that overlap, and we've cross-calibrated the two so that they're comparable as well. So that's also part of the whole process. MODIS, we improved the MODIS detection method to treat the saturation. We have a paper on that, Mishra et al., 2023, and how we approached that. It was actually a neural net fitting to Maris and MODIS as an overlap. And a key thing with MODIS is it also allows us to fill in where Maris might miss the bloom. Keep in mind, if we only have 1 or 2 Maris scenes from 2002 to 2012, 1 or 2 in a week, there's a real help to fill in and have that data for MODIS. So we used a MODIS time series as an augment to that to fill in that information. And the approach to the MODIS fit, we actually developed in a Lake Okeechobee in Florida, but we've tested it in other lakes in Florida and in the Great Lakes, and the same model works. We did not have to retune it for any of these other places. Olchi, of course, in 2016, significant changes in the calibration atmospheric correction over time. And of course, with 6 per week, that's a really great resource for the data set. Here's an example. This is Olchi data, as I've shown Western Lake Erie, this in Wisconsin, Green Bay, and Lake Winnebago. This is last August, and peak bloom, cyanobacteria. You can see very well dense bloom on the surface of the lake and on southern Green Bay in both, and the concentration ranges in here. These would be up in these reds would be up in the hundreds of thousands of cells per milliliter of microcystis equivalent. They may not necessarily be microcystis, but equivalent cells. Also, you can see some of the other lakes in the Fox River and basin, and some of the other lakes that are picked up that are not showing any blooms in these cases. So that kind of information that's available. So we do a time series process, and a key thing is we do a 10-day composite of imagery. The reason is using the daily imagery you end up with a series of problems. Clouds are one. Here's an example of three consecutive days that went into the composite. And this one is, of course, totally cloudy, except for a little spot in Sandusky Bay. Here, this was a windier day. This was a calmer day. And you can see how the change in surface concentration, this is a dominant microcystis bloom. And so if you're doing a daily, you're going to say you have a bad bloom here, and then you have a mild bloom here. Well, you don't have a mild bloom. You have a lower concentration measured. That would be the same problem if you're doing surface samples from a ship or a boat. You collect it on the one day, you have a high concentration. The other day, it's lower concentration. So we composite to the maximum value over every 10 days, and that addresses this change in concentration with winds and also the clouds. So composite that includes those images. You can see we're cloud free, so we know for a fact there were no blooms. There's a couple of other images went into this, but there are no blooms under these clouds here, and we're getting the actual biomass that's going on in the lake during that time. So we found 10 days is a good length of time to assure that we're actually picking up the biomass and clearing the clouds, low wind, low cloud conditions. And so that pretty well captures all the biomass that we need. And during any 10-day period, one of these combinations should pretty well capture the bloom. If we have 6 per week, as with Olchi, we're basically pretty much most of the time very likely to hit the bloom as well. There's a rare occasion when it's cloudy for a long period of time, but usually in the summer, over most of the country, it's not cloudy continuously, not like some other times of the year for different parts of the country. So we should be able to pick up those blooms. And so then we get the total observed biomass essentially during each 10-day period. We can tally that up for each of these. Now, we're assumed that Maris or Olchi is better for any 10-day period than CyanNet. So we calculate them separately, and then we compare the two results for any total biomass to do this. And the CyanoNet, where there's 30% higher, addresses any of the uncertainties. So if it is total biomass is higher than the others, then that's what we've applied to do this when we do a time series. So in a result one, I picked Lake Winnebago here just as a change. You actually have the Maris, the Olchi, and the Terra Cyanet as a matchups, and they align quite well. This is total biomass. Each one of these is every 10 days through the season, and during the winter there are no cyanobacteria in the lake. Olchi is doing a little better here, and I think actually reflects that this higher frequency tends to pick up a bit more of the bloom um, than Terra does. Typically, Terra also picked up a bit more than Maris. A little harder to see, but that's what we found in general. But a good match, 1-to-1 match on the data sets in general. So we can take those and generate time series. So for Lake Erie, the gold one was the time series we had previously. Again, keep in mind that was a running time series, so we had not reprocessed any data. The green is the new one where all the data has been reprocessed with all the methods we have. And what's particularly noteworthy is we're picking up more of the bloom for these early years with Maris than we are now. And that's a combination of both the Maris, the Modus, combining with each other the improved methods overall to better pick up these blooms. So we're now doing a much better job. It's all consistent data set. Everything's processed the same. The Modus, and I think I can show here. So the combination Modus is more likely to pick up Maris. This is reprocessing on both accounts. And I'd mentioned before, we were changing the Modus every year to try to improve the saturation. And so by 2015, we actually got it pretty close, which would reflect why 2015 now is similar to before. There's only one Olchi satellite, and Olchi was severely reprocessed from what we had then. That was pre-launch. And now we're up at, it's not surprising, this should pretty well match before and after because we had more calibration and more data coming in. But the green bar is now the new time series. So we can look at that 23 that I mentioned before was very bad at the time, but it was succeeded in 2008, 9, 10, 11, 13, 15, 17. Last 5 years have looked a little better than what we've seen in that 10 to 15 years ago period. So, and this, by the way, these blooms are affected by the phosphorus load, mostly from the Maumee River. That's the spring load. And the high bloom years were years where there was very high river discharge, which led to very high bioavailable phosphorus going into the lake from the river. So 11, 15, 13, and 17 had some of the highest discharge and loads going into the river. So I mean into the lake. So the variation here is a combination of how much phosphorus is running off the agricultural land and how much water, how much rain is falling in the spring leading to runoff. We're now evaluating different water bodies around the Great Lakes. This is just example time series, Saginaw Bay, Western Lake Erie, Green Bay, Lake Erie, so on. Publication and prep explaining this, and those data sets will be available for all those water bodies when we finish that up later this year. And just to give you just a few examples of the products for, in these cases, kind of on a peak bloom. So 2015, which was major bloom in Lake Erie, and you can see how it looks on Modus. Saginaw Bay had a bloom also. Here's what a little close-up of Winnebago and Green Bay. And you can see there's some shoreline loss right on the edge of the lake that you can end up with some impacts. Was part of the reason for having why you need the certain width. Maris at the 1.2 kilometer, this is what this looks like. This is the 2011 bloom. in October. There was still a bloom going on Lake Winnebago at that time. Green Bay obviously stopped, and Saginaw Bay also had a bloom as well. And this was going up the Ontario coast as well as the Ohio coast. And algae, we process each one differently, and you can see how much you can get out of 300 meter. A lot of detail on this 2019 bloom. And by the way, there was a mix of diatoms and so forth. Lake Winnebago, Fox River, and again, we can pick up some of these small lakes down here as well. There's a lot of detail. If someone's interested in more of those, they can go to the Cyan project as well. And some other lakes. We've done some work in Florida, of course, where we started this, and Lake Okeechobee, and you can see some large variations in blooms. Major bloom in 2005, 16, and 18 as well. A bit more intense blooms recently. And then we examined Lake Apopka and Lake George. Reason for a lot of the spikiness is there's a lot of popcorn clouds in Florida, and so we get some little inconsistencies as the lake starts getting smaller. One advantage of going through a whole time series is you can readily identify when you have a potential problem by just comparing one period to another on this. But you can see how consistent the Motus and Maris Olchi are against each other, and then Lake George, and how very different these three lakes are, I should add. Lake Apopka, by the way, has a bloom all the time. You can see the high concentrations. Lake George is quite transient, just like Lake Okeechobee as well. And these have been converted to mean chlorophyll concentration over the whole lake. We're also starting to look at we're looking at phenologies here. In Lake Erie, the blooms have been changing. And so we have here it's 2008, 9, 10, and you can see that those tended to peak in September, and kind of similar blooms in 1922, 23 really reached a peak in July and kind of hung out all the way to September. So later, they're earlier now than they were. They don't necessarily last any longer, but they're definitely starting earlier and tending to drag out for a longer period of time. You can see this kind of a plateau versus a peak. And that's part of that phenology. We're also going through that process for these. So some of the conclusions is it's possible to combine Maris, Motus, and Olchi for Great Lakes or others. We're going to be distributing our data for the Great Lakes quite soon, just later this summer. And this can capture, by combining Motus and Maris, we can do a better job of capturing data from 2000 or 2002 all the way up to 2000, up to the present. The new Motus algorithm gives a good consistent method for Motus. Given we've done this in Florida and the Great Lakes, we're confident this could work for other water bodies as well. And that method is available as well with the paper that Sachi did. Again, applicable to many large lakes, and can examine the phenology as well and apply these across. And I repeated that. And if that's our team here, I'll answer whatever questions there's time to answer. Thank you very much.
Thanks, Richard. That's great. It looks like we do have quite a few questions rolling in. I'd like to remind everybody to please put them in the Q&A panel rather than in the chat so they don't get lost. And we are going to address those after our second presentation. So up next, we have Rebecca North. And I'm sharing my screen. Here we go. She is a limnologist focusing on aquatic ecology and biogeochemistry with expertise on nutrient and phytoplankton dynamics in water bodies. She left the Great White North in 2016 and joined the School of Natural Resources at the University of Missouri, where she is currently an associate professor. Rebecca's lab group studies why lakes turn green with an interest in year-round processes. The MU Limnology Lab conducts the statewide water quality monitoring for the state of Missouri, which builds on a 49-year data set. Current projects in the lab include spatial and seasonal investigations on cyanobacterial harmful algal blooms and cyanotoxins. Again, any questions you have, we'll address at the end of Rebecca's talk. And put them in the Q&A panel. Rebecca, I'll stop sharing and the floor is yours.
Thank you, Amy. So thanks everyone for attending today. I'm going to be kind of talking around and addressing the question, should we be worried about winter cyanotoxins? So this work is, conducted out of the University of Missouri Limnology Lab. I'd like to acknowledge my co-author and collaborator, Dave Richardson at SUNY New Paltz. And most of the data I'm presenting today is the result of the master's thesis of Yusuf Olale. So kind of starting off with how we perceive lakes. So if your definition of limnology was anything what I learned as an undergrad, it is the study of lakes located within 60 miles of a major university campus, but not during the winter. So this is a bit tongue-in-cheek, but it does kind of reflect the fact that most of how we define lakes and what we know about lakes is summer data. And this is, due to logistical reasons of undergraduate student availability, but also most of the government monitoring programs are focused on summer collections of samples. But unfortunately, things are changing. And so we know that our winters are getting shorter. And so this unmonitored season is changing without having a lot of kind of baseline or background information to be able to assess that change. And if you're a real pessimist, you'll look forward to the future and think that most of our lakes will end up being ice-free by the end of the current century. And so we often think about, harms and risk to harmful algal blooms occurring when you're swimming or, recreating in the water. But these are actually year-round potential human health risks. So both people and animals can be exposed to the toxins that cyanobacteria produced through the ingestion and inhalation of aerosolized toxins. So this could be, boating on the water or even living or walking around a lake. So you don't actually have to be in the water to be exposed. They can actually enter the air. They can also enter the food web. And so, you'd have to eat a lot of fish, but it is potential to have contamination in fish tissues that are consumed by humans. And of course, we drink our water year round. So if your drinking water source is surface water, you potentially can be exposed to toxins that way, though most of our drinking water processes effectively remove most of these toxins if they're aware of them. So the objectives or the format for today's talk is to address the following three questions. So do we get winter cyanotoxins? Should we be concerned? What are some of the drivers of these cyanotoxins? And then looking to the future, what will happen in the winter and these year-round processes related to the production of cyanotoxins. So most of the data that I'll be presenting comes from the state of Missouri. So if you're unfamiliar, I like to refer to it as the middle of the middle of the middle, or the flyover zone. So it has become popularized in the Netflix show Ozark, which features one of the study sites that I'll be talking about, Lake of the Ozarks. This is a unique set of water bodies. So there's no natural lakes in the state of Missouri. So they're only reservoirs. And there's a really nice, well, I think it's nice as a scientist, but trophic status gradient from north to south. So the north of Missouri is primarily agricultural land use. So we tend to have really kind of green or eutrophic lakes in that region of the state, whereas the south is 60% forested. So kind of natural land use, and most of the lakes in that part can be oligotrophic or really clear in visibility. So given this nice gradient and trophic status, we can use this study system to address a lot of research questions. And unfortunately, it's not hard to find cyanobacteria. So the data that I'll be presenting on came from primarily two programs. One is the Statewide Lake Assessment Program, or SLAP. And so this has been consistent statewide water quality monitoring for 49 years. The data that I'm presenting today is from 2017 to 2023, so 7 years worth of data, and we sample 65 reservoirs a year. It is a summer-only sampling project, so we only sample during summer stratification, and usually about 4 times. And the samples all come from the deepest point in the reservoir, which is in front of the dam. This is supplemented by year-round sampling conducted primarily by high school students. So this is a project called ROSS, Reservoir Observer Student Scientists, where we get students to walk out from their high school to their local reservoir, collect a water sample on a weekly basis, and then they, as you can see here in the picture, filter it and process it in their classroom and then freeze it, and then the samples all get analyzed in the MU Limnology Lab. So this was originally piloted and funded by the North Central Region Water Network and is currently funded by the USDA. So as I mentioned, the unique part about the state of Missouri is we have a full range in trophic states. So down here you can see we've got lakes representing oligotrophic, mesotrophic, eutrophic, and hypereutrophic, where you can kind of think of this as a range from, kind of clear blue waters to green waters. We monitor for 4 different cyanotoxins, and so this top graph here is showing anatoxin concentrations. The second one is cylindrospermopsin concentrations. The third one is microcystin concentrations, and so this is probably the most popular toxin that you've probably heard a lot about. And then finally, we have saxitoxin concentrations. So going into this work, the hypothesis would be that the green lakes are the ones with lots of nutrients and chlorophyll and potentially dominated by cyanobacteria. So we classify these as eutrophic and hypereutrophic, would have the highest cyanotoxins. And so if we look through these different cyanotoxins, you can see here that the statistics on this, if they're not different from each other, they have the same letter. And if they're statistically different, then they have different letters. So for anatoxin, you can see that this hypothesis is true, that there's no difference between oligotrophic and mesotrophic anatoxin concentrations, significantly higher in eutrophic, and then significantly higher again in hypereutrophic lakes. However, for cylindrospermopsin, it doesn't follow this pattern at all, and in fact, the opposite is true. And so here we have the highest cylindrospermopsin concentrations happening in our oligotrophic or our clear lakes. Microcystin follows kind of the hypothesized pattern, with the highest concentrations happening in the hypereutrophic and eutrophic lakes, as with saxitoxin. So already just based on this first initial analysis, you can see that not all cyanotoxins behave the same way. And so this is some of the complicating factors when we start to try to manage these systems, because we're not just managing for one toxin, we're measuring for a variety of them. I'll also mention that these toxins were analyzed using ELISA kits, and so we don't know the congeners of these different toxins. These are just total concentrations. So looking at these concentrations of toxins, again, anatoxin at the top, cylindrospermopsin, microcystin, and saxitoxin, we can first start comparing them to kind of these criteria for action levels. So really, the concentration has to be above 10 micrograms per liter for there to be any kind of recreational action level. Again, we don't compare it to the drinking water standards because this is raw water collected right from the lake itself. And of course, drinking water gets treated before consumption. So it's good to see that we don't have any concentrations that exceeded the action levels. So the concentrations we're talking about here are all, pretty low in the relative manner. However, they follow, again, very different patterns. So looking at anatoxin first, you can see here that it's pretty predominant in the summer with the highest concentrations in the late summer period. Cylindrospermopsin is the lowest concentration, so most of the time it's below detect, but when it is high, they're mostly non-summer peaks, so happening outside of that summer season. Microcystin is pretty persistent year-round, and the peaks are occurring in the fall period, so again, outside of that summer period. And saxitoxin is mostly in summers with late summer peaks. So already we can see that the when and the where of when to expect concentrations of these different toxins differ by toxin. So to address the first question, do we get winter cyanotoxins? The answer is yes. This is the same data, but showing kind of the smoothed data line with the median concentrations. So I kind of presented it this way to kind of suggest that maybe our summer-only sampling is not sufficient to capture this type of variability. So if we look at all of our typical summer sampling season from June to August, this is the picture or the dynamics that we would capture. And so you can see here we're missing a lot of kind of high concentrations and a lot of variability outside of that summer sampling window. So what is the parameters that might be driving, or when and where would we have high cyanotoxin concentrations? Well, I won't bore you with the details, but we did GAM analysis on a variety of different parameters. So we kind of combined them to talk about them as morphometry. So these are things like latitude, longitude, watershed area, etc. We looked at lake physics, and so this is the light available to the phytoplankton, the Secchi depth, the mixing depth, things like that. For water chemistry, we looked at total nutrients, so nitrogen and phosphorus, and their ratios. We looked at the forms of nitrogen and phosphorus, as well as TSS, chloride and silica, and DOC. And then for phytoplankton, in addition to looking at the toxin concentrations, we also looked at the chlorophyll A concentrations, which we typically use as a proxy for phytoplankton biomass, particulate organic matter, and then we did phytoplankton counts on a subset of the samples, looking at phytoplankton and cyanobacteria biovolumes. And so here I'm showing numbers for each cyanotoxin of the percent deviance explained. So the highest numbers shown here explain the most variability in the cyanotoxin concentrations. So you can see for anatoxin, all four kind of categories explain some of the deviance in the data, with the highest being 23% was explained by water chemistry. For Cylindrospermopsin, only two of the four categories showed any kind of significant explanation, and that was both for lake physics and water chemistry equally. For microcystin, there was hits in all four categories, with the highest being kind of a three-way-ish tie, with water chemistry explaining the most, 19%, followed by lake physics and phytoplankton explaining 13%. And then finally, saxitoxin is across all four categories with the highest percent of deviance explained for the morphometry, followed by lake physics and then water chemistry and phytoplankton. So already, again, we can see that this issue has gotten complicated in that different toxins have different drivers. Looking at these a bit more specifically, for anatoxin, again, the water chemistry explained about 23% of the deviance, and that was mostly related to total suspended solids and silica concentrations. Again, when you look at these GAM analysis, hopefully you can see that these are all non-linear relationships, so a little hard to make predictions. Looking at Cylindrospermopsin, we can see that 9% of the variance was explained by water chemistry, and this was dominated by total nitrogen, the ratio of nitrogen to phosphorus, and total suspended solids. Lake physics also explained to 9%, and this was dominated by Secchi disk depth, the depth of the dissolved oxygen maximum, and the change in density, so like a stratification parameter. For microcystin, it kind of had three major categories of drivers. This included 19% was explained by water chemistry. So this is total nitrogen, total dissolved nitrogen, and dissolved organic carbon. For phytoplankton, it explained 13%, and this is particulate organic material, chlorophyll A concentrations, and pheophytin concentrations. And then finally, lake physics explained 13%, and that was primarily driven by KD, or the attenuation coefficient, an indicator of light, surface pH, surface dissolved oxygen concentrations, and the anoxycline, so a point in which oxygen disappears. For saxitoxin drivers, the strongest explanatory variable was morphometry, and this was the watershed area, the latitude of the water body, and the perimeter of the watershed. So yeah, hopefully just that scan through things allowed you to appreciate that we have a variety of different drivers and they react differently for each of the different cyanotoxins. So most of our management of these lakes and our public messaging is to, scan before you swim. If the water appears green or has a lot of phytoplankton in it, that it could potentially be toxic. But as mentioned, we did look at a variety of phytoplankton parameters to try to test this theory, and we were only able to explain just over 10%, and that was only for the toxin microcystin. So perhaps our indicators of, kind of toxic lakes or things to avoid might be failing us, and we should really consider these proxies and also, investigate the fact that they are all non-linear relationships. So in this case, the devil might be in the details. So to answer the question, what are the drivers of cyanotoxins? Unfortunately, it's different for each cyanotoxin, and no one driver explained more than 24% of deviance in the concentrations. And so we still haven't got, a magic bullet solution to one size fits all for these problems. Thinking ahead to the future, what is this going to look like? And thank you, Rick. You already gave me a prelude on this where you said, yeah, the phenology or the peaks of at least chlorophyll A concentrations seem to be changing. This fits nicely with our hypothesis. So we're in the first year of a new NSF project called Thin Ice. So myself and 4 other collaborators are looking at kind of winter futures under climate change. And so this is kind of our conceptual diagram. So for northern temperate lakes that have ice and snow in the winter, as they continue to lose this ice and snow, then you can get this intermittent ice cover. And so this is where things kind of freeze and then they melt and then they freeze again. And finally, in warmer latitudes, like where I am in Missouri, we don't get ice cover that often, and so we end up getting the whole water column mixing from top to bottom. And so we're using a space for time approach for this. So we're looking at a variety of lakes across a variety of latitudes and ice cover to try to see if we can make predictions along these lines of climate futures. So we anticipate that as we lose ice, it'll change the physical, chemical, and biological environment of our lakes. So here we're kind of showing our theories on this. So current kind of situations or lakes that are ice covered are called dimictic lakes. So those are represented by the gray. And then the black lakes are the ones that have no ice cover. So they tend to be kind of mixing, at least in the winter months. And those are referred to as warm monomictic and represented by black. So here in the future, we expect as lakes kind of warm and we lose our ice cover for them to get warmer surface temperatures. We expect changes in the mixing dynamics, particularly in the winter, because it is that ice cover that helps to have convective mixing. So it keeps the phytoplankton up and suspended high on the water column. So in futures, when we lose that ice, they're going to get mixed down deeper in the lake. And this is going to impact the mean light or the amount of light available to the phytoplankton. And it will really depend on how deep the lake is as to whether or not this will cause them to be light limited. And we also expect that this will cause changes in the surface nutrients, so looking specifically at nitrogen and phosphorus. And so currently in our lakes, we mostly see our cyanobacteria shown here as the cyan color, kind of in that late summer peak. But again, as Rick alluded to, we expect in the future to see that peak to get a little more delayed and a little more sustained. So kind of more dominance of cyanobacteria on a year-round basis. So to circle back to the original question, should we be worried about winter cyanotoxins? Unfortunately, yes, we do get winter cyanotoxins. They're already a thing. And the drivers are not entirely clear. There's not one thing you can change in your lake that could maybe prevent them. They seem to be variable, and they also depend on which type of cyanotoxin you're concerned about. And so what will happen in the future, we expect to see more year-round cyanohabs. So this is a problem that's not going to go away in the future. And so, thinking about a monitoring perspective, perhaps it's time to revisit our kind of summer-centric lens and start trying to expand our monitoring programs to include all months of the year to be able to capture these changing phenology and changing dynamics. So with that, I'd like to acknowledge all the funders of this project and welcome any questions anybody might have.
Thank you, Rebecca. I'd like to thank everyone for participating in the Algal Bloom Action Team's webinar series. A special thanks to Rebecca and Richard for sharing. Rebecca, if you want to stop sharing, then I can put up contact information for each of the presenters. And I believe you can reach out to them directly with any questions you have that we don't get to during the end of this webinar. And a special thanks to Ann Nardi and her team at the North Central Region Water Network for hosting the Zoom webinar and coordinating the technical aspects of the webinar. As we move into our Q&A session of the webinar, I'd like to remind you to check out the Algal Bloom Action Team's website, which again, I'm putting in the chat. And here you'll be able to connect with the team and subscribe to our events. So moving into the Q&A, we have several questions here. Let's see. Rebecca, it looks like you're still sharing.
Oh, sorry.
That's okay.
Close that.
All right. So Robert has a question, I believe, for Rick. How deep can you measure into the water to get a signal?
Rule of thumb is a Secchi depth.
Okay.
So it's the surface concentration there.
And what wavelengths or bandpass regions are measured for algae and cyanobacteria?
We use on the sensor, what's available on the sensor, we use 620, 665, 681, 709, and 754. And that allows us to pick up characteristics of chlorophyll A absorption specific to cyanobacteria and also phycocyanin.
Right.
And with it being the way we do it is quite insensitive to sediment. which can be a problem for some methods of doing this.
Are the satellite images available to anyone?
Yeah, the Cyan project has, and ours, we are rolling ours out available either our website or through NCEI, NOAA's Climate Observatory. So more will be for the newer ones for the Great Lakes will become available later this year. But yes, they are available. I can get you contact information.
Great. Gina says, thank you for mentioning the effect of wind speed on surface scum formation. I appreciate the inclusion of the wind speed and direction arrow on your Green Bay and Winnebago product, which helps me to explain bloom dynamics in public or to the public. So thank you for that. How do you measure discharge loads from the river? Do you have stream sensors or take nutrient samples?
We don't, but we work with Heidelberg University, the National Center for Water Quality Research, who has automated sampling for water quality for the nutrients, and then they put those at USGS gauging stations. So that makes it possible to put this together. There's an amazing network they have in Ohio and Southern Michigan.
We've got some questions for Rebecca here. Can you speak a bit more on how cyano represents microcystis equivalents? We at the UW Oshkosh Group NSF project with Tim Wine are studying HABs in Lake Winnebago and often see, I'm going to stumble over this, a phenomenon dominance. Do you have an algorithm to convert phycocyanin in that species to phycocyanin and microcystis?
I'll try to get this right. It was a complicated question. So phycocyanin is a pigment that multiple species of cyanobacteria produce. And so I didn't show that data, but we do measure phycocyanin through sensors. So we have some profilers that have a sensor on it, XO3s. And then we also actually have a method in the lab where we filter them onto a filter, and then we can extract the phycocyanin concentrations similar to what we do for chlorophyll A concentrations. But I didn't present that data. So those would represent, cyanobacteria, but it might not represent the cyanobacteria that are producing toxins. And so they're really complicated. There's only certain types of cyanobacteria that produce toxins, so you shouldn't assume that they're all producing toxins. And even if they have the ability to produce toxins, they can turn it on and off. So that's really the key question is we have no idea why cyanobacteria produce toxins. Because if we could figure that out, then we could start, playing with that switch. So there's a lot of assumptions to be made in your question. And so I think, definitely measuring the phytoplankton themselves might be the best approach.
So Robert says that question was actually for Rick.
Okay, good. That's why it was so complicated for me.
I think your answer was a better answer than I would have. We probably need to talk about your data, actually, given you're doing
and I've seen some questions there. And so unfortunately, it's only a master's thesis at this point, and Dave and I are working on publishing it. So it's not submitted or published yet.
Yeah. But yes, I think I'd like to talk to you more about what you're doing.
Yeah, ditto.
One of the things in these seminars is
Great.
making connections.
Rebecca, do you have any data on which species are producing cylindrospermopsin in your oligotrophic reservoirs? Gosh,
yeah. So
tough.
it's mostly cylindrospermopsis, which is types of cyanobacteria that can produce it. That's the other complication is some cyanobacteria can produce multiple toxins. So there is some cyanobacteria that can produce all 4 or 3. So it's not, a 1-to-1. If you have this, then it must be produced by this. So in our case, we are southern enough, and we are starting to see a northern spread in the species cylindrospermopsis, which is primarily producing that toxin.
Thank you. Rick, how is the morphology of the organism connected to your signals? Is this done occasionally or all the time?
The morphology of the organism?
Yes.
there's the determination of, say, maybe this is the answer, but the determination of a chlorophyll concentration from the satellite, there's a little bit of a morphological factor to that, which could change a bit from one to another species. Factor of 2 or 3 potentially on an absolute scale. The relatives would be very consistent for any given species. I'm hoping that's the answer. I'm answering the right question. I put a chat into our website, by the way, for our monitoring for HABs, and that can also start leading people to links or ways to get in touch with us for more information.
Yeah, and a special thanks to everybody who's sharing their data sets as well in the chat. That's great. Like Rick said, this is the purpose of these webinars, is to connect researchers and make a larger impact. Let's see. So you presented, I believe, Rick, some figures on CHLA concentrations that are averaged across lakes. On the Cyan web app, we can click on a pixel to see the concentration within that pixel. But is there a way to find the average across the water body?
The Cyan app? No.
Yes.
Not now. Our new project for high resolution, we're definitely working on that. But EPA, I believe in their Enviro Atlas when they process the data, so this wouldn't be live, but the Enviro Atlas at EPA, I believe, is doing lake analyses, lake-wide analyses. So that's worth a look at, the EPA Enviro Atlas. I'll have to pull up a link for that. But the Cyan data is in there, and they use the various algorithms we've done for that to come up with those for those 1,800 or so lakes in the country.
We have time for a few more here. Rebecca, is this analysis of cyanotoxin drivers published in a paper? I believe you addressed that, right? It's not yet published.
Yeah, just a master's thesis.
Mm-hmm. Just a master's thesis.
A very well done master's thesis.
How often were the samples collected, Rebecca?
Yes.
Yeah,
So
go ahead.
For the SLOP data set, it was on a monthly basis. So once a month for 4 times over the summer. And then for the ROS data set, they're weekly samples.
Okay. Yeah, Shelby was curious about the flashiness of antitoxin concentrations in oligotrophic lakes or lakes with higher. Oh, that question disappeared. Never mind. Higher or with high sunlight transmittance and thus possibly quick degradation of the toxin.
Yeah, no, that's a great point. And that's actually, I gave a seminar in the summer in this series and talked a lot about experiments we did with the degradation of these toxins. So I think that is a point to be made. I think there is a lag period between when they're produced and when we actually measure them. So in some cases, Bacillus spermopsin, for example, it doesn't degrade or break down as quickly as the other toxins, which may be why we're seeing more of it in the water column in these kind of non-summer seasons. And things that degraded are, kind of bacterial activity, so warm temperatures, UV light. And so anytime that there's less of that, you might get the toxins persisting for longer in the water column. So they're not, they could be kicking around longer than the phytoplankton that produce them.
Right. Rick, what are the prospects for using handheld spectrometry or photometry tools for smaller bodies of water?
If you have the right wavelengths or a hyperspectral sensor, it's quite doable. If you're just using a smartphone, I mean, not so much, because just because it's green doesn't answer the question of why it's green. Rebecca's reference about phycocyanin, for example, as well. But there are approaches out there for handheld units to do that. We do some work of our own. We have handheld radiometers that we use for algorithm testing and evaluation, and for small bodies of water. Also, I mean, some of these can go on drones. We're working on we've got one that's a drone agnostic sensor that you can fly in any drone, and it collects doesn't collect images, it just collects the data, and you can actually look at it real time. So we've been working on a method there to speed up the ability to determine how much cyanobacteria or chlorophyll you have.
Right. Well, we're right at 12 o'clock Central. So I've put, like I said, the email addresses, contact information for the speakers are up on the screen. Feel free to reach out to them if we didn't get to your question. And again, thank you for joining us. We look forward to the next webinar and seeing everybody again.